Hello everyone, and welcome to my Exam 3 Video 1. My name is Shayla Kim, and in this video, I will be explaining Hershey and Chase's bacteriophage experiment. Before I get into this experiment, I should give you some background information to explain why Hershey and Chase conducted their experiment. In the early 20th century, scientists have been trying to figure out if genes were composed of proteins or DNA. Initially, many scientists believed protein to be the genetic material due to its abundance and variability. DNA only had 4 building blocks called nucleotides, which scientists thought didn't offer much variability. It wasn't until a few experiments, such as Griffith's transformation experiment and Avery, McLeod, and McCarty's transforming principle experiment, that scientists began to see that DNA was essential in genetic information. It was concluded in Griffith's experiment that a heat-killed virulent bacteria transformed harmless 2R bacteria into the deadly 3S bacteria. This was called the transforming principle. Avery, McLeod, and McCarty wanted to look further into the transforming principle and conducted an experiment using heat-killed 3S and treated 2R bacteria. 2R bacteria were treated with RNAs, proteins, and DNAs, respectively, and then mixed with heat-killed 3S. The only sample that did not contain transformed 3S was the one treated with DNAs, meaning DNA is the transforming substance. In 1952, Alfred Hershey and Martha Chase performed a series of experiments on bacteriophages. Bacteriophages are viruses that infect bacteria. They knew that bacteriophages attach themselves to a host bacteria cell and injected their genetic material into it. The question that came up was whether the genetic material was proteins or DNA. To conduct the experiment, Hershey and Chase prepared 2 different batches of bacteriophage. One batch was produced in the presence of 35S, a radioactive isotope of sulfur, and the other was produced in the presence of 32P, a radioactive isotope of phosphorus. Sulfur is found in many proteins but is absent in DNA, so only bacteriophage proteins were radioactively labeled by 35S. Phosphorus is found in DNA but not in proteins, so only bacteriophage DNA was radioactively labeled by 32P. Each batch was used to infect a different culture of bacteria. After the bacteria were infected, the protein coats were sheared off, and each culture underwent centrifugation so that the virus and bacteria would separate. The bacteria, which are heavier, move to the bottom of the tube and create a lump called a pellet. The lighter materials, such as the culture medium and bacteriophages, move to the top of the tube and form a liquid layer called the supernatant. Hershey and Chase measured the radioactivity in the pellets and supernatant from both tests and found a large amount of 32P in the pellet, and most of the 35S were found in the supernatant. The results indicate that DNA is the genetic material because DNA was transferred into the bacteria, while proteins stayed in the supernatant. Hershey and Chase concluded that DNA is indeed the genetic material in bacteriophages. Thank you for watching.